All right, so in this lecture, we're going to discuss the nervous system. So this is the first lecture that's starting off with the nervous system. So we're going to talk about some of the fundamentals. And so when we think about the nervous system, as far as what does it do? Well, so the fundamental unit of the nervous system is what's known as the neuron. And so in order for the nervous system to do its thing, what it does is it'll send action potentials to each of the different neurons. Okay, and so these neurons, how they communicate with each other is by using what's known as neurotransmitters. And so neurotransmitters are these chemical messengers that will either excite the next neuron or they'll inhibit the neuron. And so that's at the microscopic level. But when we think about macro, what does the nervous system do? The nervous system gives us the ability to think, to see, to feel, and to move. So it's helping to coordinate all of these different things. And so the nervous system is divided into two different parts. So you have the central and the peripheral nervous system. So the components of the central nervous system include the brain as well as the spinal cord. And so the peripheral nervous system includes everything else. So some of those things include the cranial nerves. So the cranial nerves are found in the brain. And then you also have spinal nerves. And lastly, we also have what's known as peripheral nerves. And so just to show you what spinal nerves are, so if you look here, this is our vertebral column, and these nerves that are coming out, this is what's known as the spinal nerves. And so this part is cut, and so the cranial cavity, right, this is what this part is, and so what's coming down through the vertebral foramen, that's where the spinal cord is traveling through. So the spinal cord is traveling down, and then those spinal nerves is what's branching off. And so if we look at this model here, this is what's representing the spinal cord, and then these are what the spinal nerves are representing on this model. Okay, so this is just a cross section of the spinal cord. Okay, we'll discuss like some of the components that are here on the inside, but we're just trying to get a basic understanding first. Okay, so I've already discussed the two different divisions, and so now let's discuss the two main components that make up the nervous system. So I've already introduced the neuron, but we also have what's known as glial cells. So glial refers to glue. Okay, it's like the glue for the neurons, and so what the glial cells do is they support and they nourish the neuron, or our neurons, plural. Okay, so some of the different types of glial cells that we have include the myelin sheath, as well as microglial cells, ependymal, and then the astrocytes. So let's discuss some of the specifics of these different glial cells. So what myelin sheath is, if you look at this neuron model here, you see this yellow part here that's covering the axon. So this is what the myelin sheath is. And so the name of the myelin sheath, it varies depending on which system it is that you're talking about. So for the central nervous system, we know them as oligodendrocytes. For the peripheral nervous system, we know it as the Schwann cells. And so what the myelin sheath is helping to do is, we've identified like the basic components here of the neuron before, but let's like review that real quick. So this is the cell body of the neuron. These parts that are coming off, this is what's known as the dendrites. So you have the cell body, the dendrites, this is the axon hillock, and then the axon is the part in blue. The myelin sheath is what's wrapping around. And so if you look at the myelin sheath, because we know that that action potential is traveling down, right? And so what an action potential is, it's basically, right, all of these sodium ions are coming in, they're influxing in, and that they're propagating that action potential all the way along. But what this myelin sheath is doing, it's helping to insulate the axon. And so when I take this off, you can see all of the different multiple layers that are covering this axon. And so what the myelin sheath, it's increasing the speed that it's traveling. But if you think about it, it's fairly similar to, if I have my phone up here, it's charging, and you have the little white cord, right, that goes to it. So you could think about the wires that are on the inside of that cord as like the axon. And then the white part on the outside, you could think of that as the myelin sheath, right? Because it's basically the electricity that's running through our body. And so it's fairly similar to that whenever we like charge our phones and stuff. So yeah, that's a little bit about the myelin sheath and what we call them in different parts. So the next one is the microglial cells. And so with the microglial cells, they are what's known as phagocytes. And what phagocytes, what system they're part of is for immunity. Right? So eliciting a immune response. So we also have the ependymal cells. And ependymal cells, they are found within the ventricles of the brain. And so within the ventricles of the brain, what circulates there is what's known as cerebrospinal fluid, or CSF. So we'll learn more about exactly about the ventricles and cerebrospinal fluid, but we're just trying to familiarize ourselves with the different components here, the glial cells. And so these ependymal cells, they will line on the inside of the ventricles. Okay, so then the last one is the astrocytes. So cyte means cell. What astro we're referring to is the shape of it. So it's shaped like a star. And so the astrocytes, these are the most abundant, glial cells. And one of the specific functions that astrocytes do is they help to recycle neurotransmitters. Okay, so now we have a good understanding here of the glial cells, how they support our neuron. The next thing we need to discuss is how neurons communicate with each other. So I've already mentioned that neurons use neurotransmitters to communicate with each other, but there are two main types of neurotransmitters that are used for this. And so the first type of neurotransmitter is we have what's known as excitatory neurotransmitters, and then we also have what's known as inhibitory neurotransmitters. And so if you look at the difference between the two, what I have drawn here on the board, so this should already look familiar. We've learned the action potential, in which membrane potential becomes more positive once we get the influx of sodium. And so when there's an influx of sodium, right, depolarization occurs, and then once we reach our peak, repolarization occurs, and then once we go past resting membrane, this is called hyperpolarization. And so we've already learned one of the examples of a specific type of neurotransmitter that's excitatory. So it's what's known as acetylcholine. So we've learned that acetylcholine is the neurotransmitter that's found within the neuromuscular junction. So when acetylcholine binds to its receptor within the motor end plate, we get the flux of ions, and then the action potential travels along the sarcolemma. But now we're moving on to the nervous system. And so one thing that I will mention about acetylcholine, so I've said that it's excitatory in that it goes into the neuromuscular junction so that the muscle contractions can happen. But have you ever heard of Botox? Yeah. So the way Botox works is that it will inhibit the docking of the vesicle that contains the acetylcholine. So let's go over like what happens here within this neuron. So number one, or let me tell you this. So number 3 here, this is what's known as the synaptic cleft. Okay, so number 3 is the synaptic cleft. And then, so if this is the synapse, then number 1, this is the presynaptic neuron. And number 2 is the postsynaptic neuron. So then what travels here in this vesicle is the neurotransmitter. So for instance, if this is acetylcholine, in order to release it, we have to get the influx of calcium here. So calcium gets into the cell. Once it gets into the cell, this vesicle will then fuse. It'll fuse here at the axon terminal. And then it releases that neurotransmitter so it can bind to its receptor. And so like what I was saying earlier about Botox, so it's derived from a bacteria known as it's C. botulinum, I believe. But what that toxin does is it causes like muscle paralysis. And so what they'll do is they'll give like injections in the forehead, and so it basically causes like paralysis of that muscle. So that way you don't see like the wrinkles on the forehead, because obviously if acetylcholine can't fuse here at this part, right, to release the neurotransmitter acetylcholine, then a muscle contraction it's not going to happen. So therefore you'll have like the flaccid muscle, and so if they try to like make a facial expression like using up here, right, they can't do it because these muscles are paralyzed basically. So so that's just kind of like a clinical aspect as far as talking about acetylcholine. And so there are other examples of excitatory neurotransmitters. So one of them is what's known as glutamate. So you have glutamate, and then when we talk about an inhibitory neurotransmitter, instead of depolarization occurring, the event that happens is hyperpolarization. And so one example of an inhibitory neurotransmitter is what's known as GABA. So GABA is short for gamma aminobutyric acid. Okay, and so you may be thinking, well, how is it that the cell membrane, the membrane potential, becomes more negative? Well, if we look at what I have drawn here, so this is first an example for excitatory. So what will happen for excitatory, for instance, let's say that this was acetylcholine, right? So acetylcholine, I'm going to draw this in blue. Acetylcholine will bind here, and then from there, once it binds to its receptor here, then we get the influx of sodium, right? So the sodium will go from the outside to the inside of the cell. Membrane potential becomes more positive. So we're already familiar with that. But for inhibitory, whenever, for instance, GABA, when GABA binds to its receptor here, it's like a GABA receptor, that's the name of it. From there, we'll then have the influx of a negatively charged thing. And so this negatively charged thing is what's known as the chloride ion. So if a negative thing is entering into the inside of the cell, what's going to happen to the membrane potential? Is it going to go to the positive end or to the lower end? To the lower end. Yeah, so that's why the membrane potential becomes more negative. So if this is what happens, if there's an influx of a chloride ion, is the action potential going to be propagated to the next neuron? No, it's not, right? Because how do neurons communicate with each other? Action potentials. So thereby lowering the membrane potential, the next neuron won't receive the signal. And so one thing to kind of like that's relevant towards this, so for instance like alcohol, and also if you heard of Xanax, so both of these drugs what they do is they bind to GABA receptors. So, for instance, ethanol, so alcohol, will bind there, or for instance, Xanax, and it'll change the conformation, and then from there, we'll get the influx of chloride ions, and hyperpolarization will happen. So this is why alcohol is kind of like a mellowing effect, right? You kind of just chills you out, right? Because it's not like with stimulants where these action potentials are just propagating, but that's how it works for GABA receptors using chloride as the ion to hyperpolarize the cell. Okay, so now we know how they communicate. One of the other things I wanted to discuss is more specifically the astrocytes, like what's going on with the recycling of the neurotransmitter. And so what I have drawn here, this is the astrocyte. And so within the synaptic cleft, we don't want to have an excitatory neurotransmitter there for a really long time, because what can happen is cell death of the neuron. And so we have to have a way to kind of regulate this process of degrading it. And so for instance, when glutamate it'll bind to its receptor, it'll do its thing, but then some of it is not going to be within the synaptic cleft or binding to receptors. And so what will happen is within this astrocyte, it'll take up the excess glutamate. So number 4, so that's what I have right here. This is the astrocyte. So it'll convert glutamate into glutamine. And so both of these are just amino acids. Right? Amino acids make up proteins. And so at the end of glutamate, it looks like this. It has this carboxylate ion. And then the R group, this is where the carbon skeleton of the amino acid would be. For glutamine, the only difference is this. It has this amine group attached to this carbonyl. So this is the only difference between the two. And then, so it'll recycle it and then get it back here into the presynaptic neuron, so we have enough of the neurotransmitter. Okay, so this is good for the fundamentals of this. So now let's move on to the spinal cord. So I've already showed you the spinal cord and some of the different parts within the spinal cord. So this is the white matter, and then this part on the inside, this is what's known as the gray matter. So let's write that here on the board. So what I have 1, so 1 is here on the inside, right here. So this is the gray matter. The part that's on the outside, this is what's known as the white matter. And so some of the different components, like what's going on here. So E, what I showed you earlier, which is right here, this is what's known as a spinal nerve. And then this part that's here on the back side, this is which is labeled as D. This is what's known as the dorsal root ganglion. So you have the dorsal root ganglion, which is, if you look at here on this model, it's this part right here. Okay, so now that we've identified some of the basic parts of the spinal cord, let's discuss a little bit about classifying neurons. So I'm going to move this, put this here. So we can classify neurons using a couple different categories. So the first is structure, and then the second is what we know based off of its function. And so the 3 main types for its structure, what we have, this first one here is what's known as unipolar. This one is bipolar. And this is multipolar. And so we can categorize them based off of what is coming off of the cell body. So because there's only one thing that's coming off of the cell body, we call this unipolar. Because there are 2 things coming off of the cell body here, we call it bipolar. If there are multiple things coming off, we call it multipolar. Okay, so this is classifying them based off of structure. The next is based off of their function. So we have what's known as motor or efferent neurons. And then we also have sensory or afferent neurons. These terms are used synonymously. And how we go about categorizing them, whether it's motor or sensory, is based off of the direction that the neural impulses or the action potentials are traveling. So if it's sensory, so when I say my senses, what do you think of? Like vision, right? You think of hearing, think of touch. So an example, so I have here, this is the skin model, and so we've already identified like some of the basic parts here, like all these different nerves and stuff that are responsible for touch. So in order for me, so when I put my hand on the table, in order for me to feel that sensation, I need my sensory neurons. So the sensory neuron, that information will travel from my fingertips, and then the axon will travel all the way until it gets here into the spinal cord, and then it'll synapse with the next neuron. So let me show you what I mean by that. So I've already identified that this here is the spinal nerve. So then what's within this spinal nerve, if we look at this part here that's in red, so this is what would be a sensory neuron. Let me draw that, write that out. So it'd be a sensory neuron. And this specific type, what if we classified this one, what would it be out of these? Unipolar, right? Based off of there's only one thing coming off of the cell body. So the specific type of sensory neuron that's found here, so this is unipolar. So this is what's known as an inner neuron. So the inner neuron is found here within the gray matter. So we have an interneuron. And we can categorize an interneuron as multipolar. And then the last one that I have here, this is what's known as a motor neuron. So let me get this over here. So this is a motor neuron. And if we categorize this one, this is what's known as a multipolar motor neuron. So once again, we're classifying the neuron based off of the direction that the signal is traveling. So if it's going to the central nervous system, we say that it's a sensory neuron. So this one is going to the central nervous system. What are the components of the central nervous system? The brain and the spinal cord, yes. So it's going here, but then this neuron is going away. So this is what's known as a motor neuron because it's going away. And where is this motor neuron traveling to, or this efferent neuron? For the muscle. So if we look at this neuron model here, this is what's known as a motor neuron, right? It's multipolar. We can see all of the dendrites here coming off of the cell body. The axon is traveling along. And then what's depicted here, this is what's representing, it's going to the neuromuscular junction. So this here is what would represent as a motor neuron within the gray matter of the spinal cord. Okay. So now that we understand the spinal cord model, the regions, and how to classify, I also want to discuss with you, so why is it that gray matter is gray and white matter is white? Well, I've already mentioned that the myelin is what wraps around the axon. And so within the white matter, this fatty substance, the myelin sheath, that's what gives the spinal cord that white color. Okay? All right, so now the next part is discussing the organization of the different neuron components. And so whenever we say a cluster of cell bodies, what I'm referring to, what I mean is that there is a bunch of them. That's what a cluster is. So there's a cluster of cell bodies. But the names are different depending on the location. So within the central nervous system, we know it as nuclei. If it's in the peripheral nervous system, we know it as the ganglia. And so if you look over here, you'll see that the dorsal root ganglion. So ganglion is what we're referring to here, because this is within the peripheral nervous system, because it does not include the brain or the spinal cord. And so at this part, the dorsal root ganglion, is there going to be one single sensory neuron, the cell body, found there? No, there's not, because what's the definition of ganglia? A cluster of cell bodies in the peripheral nervous system. Okay, so now let's move on to the myelinated axons. So within the central nervous system, we call a bundle of myelinated axons, we call them tracts. And so these tracts are found here within the spinal cord. And so we're going to learn some of the specific examples. So for instance, the spinothalamic tract, that is a specific type of tract that transmits information. But then in the peripheral nervous system, we call them nerves. And so we're going to learn later on the specific names of some of these nerves. So to give you an example, so for instance, the radial nerve or the ulnar nerve, right? Radial is on the thumb side, ulnar is on the pinky side. And so the definition for these, right, a bundle of myelinated axons, if it's in the central nervous system, it's tracts. Peripheral nervous system, we call them nerves. And you can also, the arm and the leg models, you can already see the different nerves that are found there. Okay, so last topic. Last topic is the connective tissue. So I've already mentioned that the myelin sheath is what wraps around the axon. Well, along with that wrapping of the myelin sheath, there's also another layer of a single axon, which is known as the endoneurium. So this should sound familiar whenever we talked about the endomysium. So the endoneurium is what covers the axon that's over the myelin sheath. And then we also have the perineurium, which is where we have fascicles. So fascicles is just a bunch of axons that are together, like this, right? So let's say this was one axon, this was two, this was three. If we had a bunch of them, that's what would make up a fascicle. So then whenever we have a bunch of fascicles, so I'm just going to write bundle of fascicles. This is the part, so the epineurium is the part that covers the nerve. So earlier, right, I showed you the spinal nerves that are coming out through here. So the part that's wrapping around it, right, that is the part which would be the epineurium. Okay, so are there any questions? Okay, that's going to do it for this lecture.